Hey everyone, Miss Calabrese here. Welcome back. So in this video, we're going to be continuing our discussion of the immune system and talking more specifically about the second line of defense. All right, so when we're talking about second and third lines of defense in general, we're talking about the study of immunology. So this is the study of all of the kind of internal body responses to infection. Right. In order for you to have a healthy functioning immune system, your body has to be able to do three key things. We need constant surveillance, so we need to be looking all over the body for a possible presence of pathogens. We need to be able to recognize the difference between foreign material and self, so we need to know the difference between self and foreign. And then we need to be able to destroy what we determine as foreign. So those three things: seek, recognize, destroy. So that's what we need to do to have a healthy functioning immune system. All right, so in order to dig a little bit deeper into the immune system, first we need to understand the different players involved here. So we're going to talk about your leukocytes here briefly. So leukocytes is another word for white blood cells. And so we're going to divide all of your leukocytes into two major categories, and those are going to be the granulocytes and the agranulocytes. So granulocytes are going to be cells that have tiny little granules in them, and agranulocytes will not have those granules. All right, so here are your granulocytes, and the granulocytes are neutrophils, eosinophils, and basophils. So starting at the top, we have your neutrophils. These are the most common of the white blood cells. So at any given time, around 65% of your circulating white blood cells are probably neutrophils. You can tell a neutrophil by looking at it because they've got these multi-lobed nuclei and these tiny little granules inside. Eosinophils are the next one pictured here in the middle. The eosinophils are you can tell an eosinophil by looking at it is because most of the time they stain with these reddish granules. These guys are good for worm infestation. So if you have a worm infection or allergies, so these are really helpful in the allergic response. And then the last one at the bottom here, these are basophils. Basophils tend to have kind of like a darkerish, bluish, purple staining granules. The granules inside of basophils contain histamine and heparin. So when those granules are released, the histamine and heparin creates kind of like an itchiness, redness in the area. And these are usually the least common. So these tend to be the hardest ones to find in your bloodstream, right? Whereas neutrophils being the most common, those are the highly phagocytic cells, so that are usually kind of the first white blood cells to the scene of an infection. So if you say get a paper cut or something, the neutrophils are going to be the first ones that are coming to that area to try to phagocytize or eat any pathogens that enter in through that wound. Okay, and then the other category here of white blood cells are the agranulocytes. So agranulocytes are the ones that don't have granules. So these are things like monocytes, which is pictured on the top here. Monocytes are very large white blood cells. They're called monocytes when they are circulating in your bloodstream, when they're in the circulatory system, but they're capable of leaving the circulatory system or exiting through a little wall in the capillary. When they leave the circulatory system and enter into the tissues, we refer to them as macrophages. So monocyte in the blood, macrophage in the tissues. And then this one pictured on the bottom here is a lymphocyte. So you have two major types of lymphocytes, your B cells and your T cells. And these are going to be really important for your adaptive immune system or the acquired immune system, the immune system that's important for specificity and memory. Also, natural killer cells are another member of this agranulocyte community. These guys are are derived from the same cell line as the lymphocytes, but the natural killer cells are not a specific part of your immune system, so they're more part of your innate immunity than the specific immunity. Okay, all right, so this innate immunity, or this second line of defense, so these are internal innate barriers. So this is what happens after a pathogen makes it across that first line of defense. If you remember, the first line of defense like skin, mucous membranes, lysosome, pH, all that kind of external barriers to entry. Once something makes it inside, then it's going to encounter one of these second lines of defense. So the first one there, and probably the most crucial, is phagocytosis. So that means phagocytosis by these different white blood cells that are capable of eating pathogens that they encounter. And so that phagocytosis is mainly going to be the job of neutrophils and macrophages. They're the ones that are going to be doing most of that phagocytosis. And another thing here, inflammation. So inflammation is a critical part of your second line of defense. Fever is also going to be critical to protecting you against infection, as well as complement interferons and natural killer cells. And we'll talk about each one of these one at a time. All right. So starting with phagocytosis. So again, a really important process for your immune system. In order for phagocytosis to happen, usually we need to get the white blood cell, whether it's a neutrophil or macrophage, to the site of damage, which means it has to leave the bloodstream. It leaves the bloodstream through a process called diapedesis. So long word, but it means basically just oozing out of a capillary and into the surrounding tissue. So these cells have to be able to undergo diapedesis, and then once they get to the site of damage, then they can ingest any pathogens through endocytosis. So, and remember, phagocytosis is a type of endocytosis where we're bringing in a large outside particle. So in this case, in this picture, what's being depicted here is a bacterium is being ingested through phagocytosis. Now the bacterium is inside the cell in a little vesicle called a phagosome. The phagosome is then going to merge with a lysosome. And remember, lysosomes are the little organelles that contain digestive enzymes. So the lysosome will release those digestive enzymes into the phagosome that contains the pathogen, and that's going to break it down. Essentially, it'll just digest that bacteria or virus or whatever it happened to be. Right. Here's a little kind of picture of what diapedesis looks like. So say that there's been damage to the surface of the skin. So that the initial thing that's going to happen here is the cells that have been damaged, so the cells that have been cut are going to start releasing chemical signals so that the body knows that damage has happened, right? So they're going to release these chemical signals that will find their way to the bloodstream, and that's going to encourage diapedesis. So that's going to encourage monocytes to leach through the capillary walls and become macrophages. It's going to allow neutrophils to go on the other side of the capillary wall and find their way to the site of damage. The cells that already live here, so the cells in the skin that are being damaged, if they detect the presence of pathogens, which is almost always going to be the case, right? When you break a surface barrier, then you leave yourself open to potential infection. Those resident cells there are going to release complement factor C5a. So this is a complement protein that's going to tell these phagocytes that there are pathogens present. So this is going to initiate the phagocytic response. All right. Another thing that's going to happen after some kind of damage or infiltration from potential pathogens is we're going to trigger an inflammatory response. So inflammatory response is just going to be a reaction to damage. The main kind of characteristics of the inflammatory response are redness, heat, swelling, and pain. And so this is, although uncomfortable, this is a normal and necessary part of your innate immunity against pathogens. So the things that are basically happening here that result in this redness, heat, swelling, and pain are, one, we're increasing blood flow. So basophils in the area are releasing heparin. That's going to increase the diameter of the vessels, which is going to increase blood flow to the area. As we increase blood flow to the area, that's going to bring more blood, which brings redness. And blood also carries heat. All right, so the increase in blood flow is what brings the redness and the heat. We're also going to make the capillaries a little bit leakier. So we're going to increase the permeability of the capillaries so that we can encourage more diapedesis. And although we're encouraging diapedesis, it's not just the cells that leak out of that capillary, also plasma is going to leak out of the capillary. And so as plasma leaks out of that now more permeable capillary bed, we're going to get swelling, localized swelling. And that swelling is going to put pressure on underlying nerves, which can result in pain. Right, so that's the basics of how your inflammatory response is working. We're increasing blood flow to the area so that we can get more healing leukocytes to that area. And then as we encourage diapedesis to get those leukocytes to the area of damage, you're going to be increasing swelling, which increases pain. Right. Another kind of non-specific response or an innate response to infection is a fever. So again, fever is one of those things that's not super comfortable, but it is really useful in helping fight off a potential infection. So the typical process for initiation of a fever is when all of these phagocytes that are running around and eating up potential pathogens, as they phagocytize the pathogens, they're going to release a chemical called pyrogen, and that pyrogen is going to trigger the hypothalamus, which is a region in your brain responsible for maintaining homeostasis. It's going to trigger that hypothalamus to increase your body's set point temperature. So normal set point is around 37 degrees Celsius. When the hypothalamus receives these pyrogen signals, it's going to increase that set point. And yes, that's going to make you feel uncomfortable and kind of crummy, but it's also going to increase your metabolic rate. So as we increase the temperature, the speed of all the reactions in your body will increase, and that means we can speed up the process of creating new white blood cells and the activity that those white blood cells are doing in order to defeat the pathogen. All right, another thing that your second line of defense can do to defend you against pathogens, and this one is specific for viral pathogens, not for bacteria. But so if we can kind of remember back to the viral life cycle, so how a virus reproduces. The virus has to first attach to the outside of a cell, and then it has to penetrate the cell. Once it's gone into the cell, then it'll go through its biosynthesis step where it will take over the nucleus of the cell and get that cell nucleus to make more viral particles. So then we do the assembly and then the release of these new baby viruses. So that's the typical lytic cycle, right? Attachment, penetration, biosynthesis, assembly, and release. Interferon is a compound that your cells can make to defend them against viruses. So basically, what happens here is once a cell has been infected by a virus, that cell can produce interferons, and these interferons will be released from that cell, and they're going to act as a warning signal to neighboring cells. So the first cell, the one that was infected with the virus, here the one on the left, that cell is already infected, right? There's nothing in particular that cell can do now to defend itself against the virus. But what it can do is produce interferons that are going to act as warning signals to nearby cells. So it's kind of like there are pathogens in the area. Let's lock the doors and turn off the lights and hide, and basically don't let that virus into your cell. So that's what an interferon tells a neighboring cell. It says, okay, we have to watch out for this. We have to prepare for this because there are viruses in the neighborhood. All right, so that's the role of interferons. All right, your complement system is another aspect of your second line of defense or internal innate barriers. Complement is a series of proteins that are in your bloodstream, and they don't do much on their own. They're basically just floating around in the bloodstream, not causing any problems, but not doing you any favors until they're warned by antibodies of the presence of a pathogen. So antibodies can trigger these complement proteins to essentially poke holes into the surface of a bacterial cell. And once they poke holes into the surface of that cell, that'll lead to the overall destruction of the cell. So complement proteins are these kind of dissolved blood proteins that can act only in response to antibodies telling them to target a particular cell for destruction. All right. And the way they work is by forming what's known as a membrane attack complex. So this membrane attack complex basically is this. It's a pathway that's going to result in building these holes, basically these perforations in the surface of that bacterial cell. And so as we create these perforations, we mess with the osmotic balance of the cell, and that'll result in cell lysis eventually. All right, and the last aspect here of your second line of defense or your internal innate barriers are natural killer cells. So natural killer cells, these cells they look a lot like lymphocytes, and in some ways they act a little bit like lymphocytes. Basically what they can do is recognize when your own cells have been compromised. So they can tell when one of your cells has been infected by a virus, and they can go kill that cell before the virus has time to spread. They can also recognize when your cells become cancerous. So if a cell mutates and becomes cancerous, natural killer cells can usually distinguish that and go destroy that cell before the cancers spread. So a very important part of your innate immune system. Okay. All right. So key to this whole second line of defense working, especially the phagocytosis part, is this recognition of self versus other. So you have to be able to distinguish between your own body cells, things that are supposed to be in your body, and foreign cells or viruses or any kind of foreign material. So we need to distinguish between self and non-self. All right. And the way that this works, the way that all of your immune system works is basically by cell surface proteins. So we need these proteins on the surface of our cells to act as sort of kind of recognition, or you can hear them referred to as like ID, like showing ID to prove that either you're part of this body and you belong here, or you're a foreign invader and you do not belong here. All right, so one of the ways that your innate immune system works is by recognizing the presence of pathogen-associated molecular patterns. These pathogen-associated molecular patterns are basically proteins that are found on the surface of pathogens that are not going to be found on the surface of your own cells. So when your white blood cells, like macrophages or neutrophils, are running around looking for pathogens, they're going to basically stop every cell they run into and say, okay, are you self? Are you foreign? Let me see some ID. And so they'll feel around for these cell surface proteins. And if they recognize them as being self, then they leave that cell alone. That's what's happening in the top picture here. In the middle picture, this leukocyte has found a foreign object, a pathogen here, who's got a pathogen-associated molecular pattern on his surface. And once he recognizes that PAMP, he's going to ingest that bacteria, right? So I want you to keep in mind that this is a non-specific phagocytic response. That means that this particular cell here, that this phagocyte does not know, he doesn't know if that's E. coli or Staph aureus or tuberculosis. He has no idea what it is. He just knows it doesn't belong here. So he can recognize the pathogen-associated molecular pattern and say, okay, that's a pathogen. I'm going to eat it. So all that does is let you determine between self and foreign. It is not a specific recognition of what that pathogen actually is. Okay, so all of this system is based on these surface markers. And the surface markers are usually proteins, sometimes glycoproteins, so a combination of like a sugar and a protein molecule that helps your immune system distinguish who belongs in the body and who does not. All right, so quick questions. What are your second lines of defense and what's the goal of those? And then what are the symptoms of inflammation? All right, so your second lines of defense, the 6 things that we consider to be internal innate immunity are phagocytosis, inflammation, fever, production of interferon, the complement system, and natural killer cells. Right, and the goal of all of those things is to sequester any pathogens that have made it past the first line of defense. and to introduce those pathogens to the third line of defense, which we'll talk about in the next video. And then the symptoms of inflammation, those four kind of hallmarks of inflammation are redness, heat, swelling, and pain. All right, so I hope this was helpful. Let me know if you have any questions.